actually is an enzyme? So one common question that I believe, is literally the first thing that might come into anybody's mind is, what actually is an enzyme? Right? What is enzyme? Now, a lot of the basic definitions everybody must have heard of, that enzymes are catalyst, all of that. But let's first talk about what actually are enzymes. Enzymes are proteins. This statement is really, really important. You know why this statement is really important? Because later on, I'm going to do the topic of protein synthesis with you, right? I'm going to talk about how the DNA is used to make proteins. So anything that goes wrong on the DNA will affect enzymes. And you can link the information together by remembering that enzymes are proteins. Enzymes are proteins that act as biological catalyst. So they are proteins that function as biological catalyst. But what are biological catalysts? Biological catalysts are substances. A catalyst is a substance that increases the rate of reaction and this is the second statement is important, and is not changed in the reaction. So enzymes are quite simply put. If anybody ever asks you, what do you think an enzyme is? An enzyme is just a protein, like all the proteins in our body, like collagen, like keratin, like our muscles, everything. It's all protein, right? So just like everything else in our body, enzymes are just a protein. It's just that they have a very specific role. The role of enzymes, the role of protein enzymes, is that they have to help speed up the reaction. They will make the reactions of our body happen quicker and faster, right? And they will not change in the reaction. Yes, they will participate in the reaction, but they will not change. They're not going to become something different. They're not going to, end up becoming anything separate than before. They will remain exactly the same, right? In the reaction, the enzyme will not change. It will remain exactly the same, but it will help make the reaction go faster. So it is simply a protein, and the role of that protein is to make sure that it makes everything go faster, right? And when we talk about our protein or our enzyme is, we can use it again and again and again and again and again. Why? Because it does not change in the reaction. It does not change in the chemical reactions whatsoever. It will remain the same. So I can use it once, I can use it twice, I can use it 3 times, 4 times, 5 times, again and again and again and again. Now, when we talk about enzymes, we can have two type of enzymes. We can have catabolic enzymes. These enzymes are the ones that students seem to actually know about, right? This is the kind of enzymes that they normally know about. What are catabolic enzymes? Catabolic enzymes are enzymes that break down compounds, okay? That break down large compounds into small compounds. And in your syllabus, you guys will find a lot of examples of catabolic enzymes. What are some examples of catabolic enzymes that you guys do need to know? Some of these, Hajra, you and I have already done, right? I hope they're familiar for you. We have, for example, we can have amylase. Amylase will break down your starch into maltose. So starch is a large molecule. Okay. Starch is a large molecule. So I will break it down into smaller molecules, that is maltose. Similarly, we can have lipase. A lipase will break down fat. So it will break down fat into smaller molecules. Or we can have protease. Protease will break down your protein into smaller molecules, right? So these are all catabolic enzymes that actually break things down. Then, now, there are also anabolic. The opposite of this, we can also have anabolic enzymes. Normally, when we think about enzymes, for some reason, we always think about enzymes breaking down things. But we can also have enzymes that make large molecules, that make large molecules from small molecules. Right? We can have enzymes that take large molecules and it takes small molecules and it actually goes ahead and use them to make large molecules. Now, you don't really have to worry about anabolic enzymes too much in your course because you guys don't really have as such any examples. If I can give you an example, then an example of this is DNA polymerase. We will study about DNA polymerase in the topic of DNA and genetics. So I have not taught you this polymer yet, or we have not gone over this enzyme yet, but I will teach you guys about this enzyme and its function in the topic of genetics. But DNA polymerase helps in building DNA. That's its job. It will take the small molecules and join them together to make us a large molecule of DNA. Right? So we can have catabolic enzymes and we can have anabolic enzymes. Is everything making sense so far? Does anybody have any confusion? Any part that's not making sense? Aljora, everything all clear?
Yes.
Sarah, Hajra, everything all clear?
Yes.
Yes.
Okay. Now, one of the things that we need to talk about obviously is how does an enzyme work, right? It's a very important thing that we have to understand. How does an enzyme work? When we talk about an enzyme, so an enzyme follows a theory that I'm sure we must have all heard quite a few times. It's known as the lock and key theory. The lock and key theory, by the way, is what does the lock and key theory tells us? To understand what the lock and key theory tells us, we need to understand first of all a few important pieces of information regarding our enzyme. Okay, so every single enzyme has something that is known as an active site. Okay, every single enzyme has something known as an active site. What is an active site? An active site is the region of the enzyme that binds to the substrate. The region of enzyme that binds to the substrate is known as what? It is known as our active site. Whichever part of the enzyme will bind to our substrate. What is the substrate? Right? Substrate is whatever molecule we are working with. If I'm trying to make a molecule or if I'm trying to break a molecule, doesn't matter. The substrate is the molecule that I am trying to work on, that I'm trying to build or I'm trying to break. Now, the active site is the region of the enzyme where the substrate will bind. The important thing that we have to remember is this, that the shape of the substrate is going to match the shape of the active site. Or I should have said it the other way around actually. The shape of the, this is the substrate. So we can see that the active site, shape of active site is complementary. Complementary means that it is matching basically. Okay, we will be dealing with the term complementary a lot in our syllabus. Even when we go over the topic of DNA, we will hear the term complementary a lot. So the active site is complementary to the substrate, meaning that the shape of the substrate and the shape of the enzyme will always match. This is the reason why an enzyme is highly specific. An enzyme is highly specific. What does that mean? That it will only bind to the complementary substrate. It will only bind to complementary substrate. That's it. This is how we make enzymes exclusive. I don't want my enzyme to be interfering in other reactions of the body. If I have an enzyme that is supposed to break down proteins, I don't want it to go and start breaking down lipids. I want the enzyme to do what it wants to do. That's it. Right? So I am going to need, so the shape of the active site and the shape of the substrate will match. So the enzyme will not just go binding to every single substrate it sees, it will only bind to the substrate that matches it. So it's not going to go around, breaking down lipids. If it's supposed to break down proteins, it will only break down proteins. Why? Because the shape of the active site and the shape of the substrate are complementary. We call this property of the enzyme specificity. Okay, that the enzyme is specific only for proteins. Now, what is going to happen that the shape of the enzyme and the shape of the substrate are complementary. I was saying matching, but you can use the term complementary. It is a better way of saying it. Okay. Now, what is going to happen that the enzyme and the substrate will bind together. Okay, there's a proper process of this. You guys should know this in these steps. All the steps that I'm telling you guys, please make sure you note them down because you will be required to mention them in your exam answer. So what is going to happen that the enzyme and the substrate are going to bind together. Okay, they will both bind together due to the complementary shape. Due to the fact that the shape of the active site and the shape of the enzyme are, sorry, shape of the active site and shape of the substrate are complementary to each other. Based on this, the two of them will join together. When the two of them will join together, they will make what is known as your enzyme substrate complex. This is known as an enzyme substrate complex. The enzyme will do its job, whatever the enzyme's job is. If the enzyme's job is to, for example, break it down, the enzyme will break it down. If the job of the enzyme is to break it down, then the enzyme is going to go ahead and it is going to break it down into smaller products. right? And once it has broken it down into smaller products, they will be released from the body. Okay, so now the products are formed, the products are made, and the products will be released. And the active site is once again available to bind to the next substrate. The active site, now our active site is free. Active site is now free. So see, the enzyme remains unchanged. The enzyme will not change. Since the enzyme will not change, we can use it again and again and again and again. So I can use this enzyme for the next substrate. This is known as the lock and key model. Why is it known as the lock and key model or the lock and key theory? Because just like for every lock, you have a specific key, right? You can't open a lock with just any random key. If you want to open a lock, you need to have that exact same matching key. Just like that over here, we need to have the exact same matching enzyme and product. If the product will match the enzyme, only then the reaction will happen. Otherwise, the reaction is not going to happen whatsoever. None of the reaction is going to happen. So this is why we call it the lock and key theory. And these are exactly the steps that you have to mention, that the substrate will have complementary shape, or the active site will have complementary shape to the substrate. Since the substrate will have the active site will have complementary shape to the substrate. So the substrate and the enzyme will bind together and they will make what? They will make enzyme substrate complex. The enzyme will catalyze the reaction, and the once the products are formed, they will be released and the active site will be available to bind to the next substrate. This is how the process of your lock and theory model, or how the enzyme is actually going to be involved in breaking down, or not just breaking down, even for making any you can say even for making any product. Okay, so we have to, like I said, remember, what do we have to remember? We have to remember that enzymes are highly specific. Okay, enzymes are highly specific, and the enzymes are going to only catalyze those reactions that are uh, complementary to its active site. Is everybody clear with what we've discussed so far? Any part that's not clear? We're all good with the lock and key theory. You guys should know all of these steps, that the shape will be complementary, the enzyme substrate complex will form, the products will be released, all of this. Is everybody okay with what we've discussed so far?
Yes.
Okay. Now, moving on to our next topic that you should know in this, that is really important, and that is factors affecting the enzyme activity. Okay, this you guys really need to know because you're going to get a lot of questions on this. So and you guys will not just get direct, simple questions, you guys will get questions that will be mixed up a lot. So, what are the factors affecting enzyme activity? Now, the first factor that we have to talk about is temperature. When we talk about temperature, let's go ahead and take a look at the graph. On my x-axis, I will have my temperature. And on my y-axis, I will have the rate of reaction. Now, let's say we are all doing a really interesting experiment. Okay, we want to find out what is the effect of temperature on our enzyme activity, and this is going to this is not just important for your paper one, paper two, or paper four. It's also important for your paper six, right? So if you go ahead and you decide to perform this experiment, you would notice something really interesting. What you would notice is that the shape of the graph will look something like this. Initially, the rate of reaction will be very low. Okay. But as you will increase the temperature, how can we increase the temperature? And ideally, in experiments like this, in your paper six, try to use a water bath. Okay. We can use a water bath to help increase the temperature. uh, a thermostatically controlled water bath. What does thermostatically controlled mean? That it can be externally controlled with a computer, or you can control the temperature and you can keep it at a certain value. So if the temperature is 20 degrees centigrade, it will stay at 20 degrees centigrade. It's not going to keep on rising. Shape of a graph is actually going to look something like this. Okay. Now, we all notice something really interesting. For example, okay, we can see that initially, as the temperature increases, the rate of reaction also increases. Now, why is the rate of reaction increasing as the temperature is increasing? Why does that happen? What makes the enzyme go faster? This is what. As the temperature will increase, the enzymes will gain kinetic energy, right? Your enzymes are going to go ahead and they're going to gain kinetic energy. So, as the enzymes will gain kinetic energy, they will obviously start to move faster. This is really important. Why? Because if the enzymes will move faster, it will lead to more collisions. And collisions are the main thing, believe it or not. The collisions are the main thing. The substrate and the enzyme are colliding with each other, right? This is also true for any chemical reaction. In any chemical reaction, any reactant that we're using, they will always collide with each other. Now, the funny thing is that not every collision is successful. Not every collision will give you a product, right? But by increasing the temperature, I can increase the kinetic energy. So molecules will move faster. So there will be more collisions. Now, not every collision will give you a product, but more collisions mean more chances that we will have a successful collision, right? So we will more chances that we can have a successful collision. Successful collision mean that we get the product. Okay? So that's why rate of reaction increases. Now, notice something interesting. Notice that your rate of reaction increases up to a certain value, after which the it starts to decrease once again, right? It increases up to a certain value, but then it starts to decrease. Why? So the temperature, or I should correct this over here and write down the rate of reaction reaches a maximum value. As the rate of reaction reaches a maximum value, what are we going to say? We are going to say that we are now at optimum temperature. This is the best temperature at which the enzyme will work its very best job. This is the temperature at which enzyme is going to give us the best possible job that it can do. It will give us the best possible result. This temperature is optimum. It is just perfect, right? Then, but as the temperature continues to increase, the rate of reaction starts to fall. Why does the rate of reaction fall? The rate of reaction fall because high temperatures cause the enzyme to be denatured. Now, I'm sure you have all heard of this term before that, oh, the enzyme has been denatured. What does this actually mean? What this actually means is this, that your enzymes, they have a active site. The shape of the active site is complementary to the substrate. Now, when the temperature becomes very, very high, the bonds in the protein gets disrupted because this is what? This is a protein, right? So in high temperature, what happens? What does denatured mean? What denatured mean is this, that bonds in protein get disrupted. Bonds in the protein get disrupted, they get affected, they get broken. Disrupted is the better term. They get disrupted. So the shape of active site changes. So it is no longer complementary to the substrate. This is what denatured means. It is no longer complementary to the substrate. The enzyme is denatured. Now, obviously, if the enzyme is going to be denatured, the active site is no longer matching because the high temperature has damaged the protein bonds. What will happen? Rate of reaction will decrease. And this is exactly what we see in our graph. This is exactly what is reflected in our graph. In our graph, we can see that initially, as the temperature increases, the rate of reaction is also increasing, right? That's exactly what we can see. As the temperature increases, the rate of reaction increases. Why? Because the high temperature means that they will gain kinetic energy, more collisions. So obviously, if there are more collisions, more chances that the collisions will succeed and we will get a product. But this will keep on happening until you get to a point at which the rate of reaction becomes maximum. The point where the rate of reaction becomes maximum is the optimum temperature, the best temperature at which we're going to get the best activity of the enzyme. But if we don't stop at this point and we keep on increasing the temperature, the rate of reaction starts to fall. Why? Because the enzyme has been denatured. What does denatured mean? It means that the bonds have been disrupted. The bonds of the protein molecule, because enzyme is just that protein. The enzymes of the protein have been disrupted. Since the enzyme of the protein have been disrupted, it is not going to, the shape of the active site will start to change. The active site is not going to be the same as it was before. And as a result, it will no longer bind to the substrate and our activity is going to decrease quite significantly. So every single enzyme, by the way, has its own optimum temperature. Okay. Every enzyme has its own optimum temperature. So for example, the enzymes in our body. The enzymes in our body, their optimum temperature is 37 degrees centigrade, right? The enzymes in our body, they have a optimum temperature of 37 degrees centigrade. So every single enzyme in our body, it will operate at 37 degrees centigrade. The enzymes in bacteria, they might operate at 25 degrees centigrade, right? The enzymes in plants or whatever, they might work at different temperatures. Now, just like one of the factors that is affecting the rate of enzyme activity is your temperature, another factor that is affecting it is pH. Just give me 1 second. So another important factor that we need to take a look at is our pH. Our pH has a similar effect compared to temperature. Okay. If you're asked to draw the graph for pH, this is how the graph for pH is going to look. So on our x-axis, we have our pH. On the y-axis, we have the rate of reaction. Let's say our pH is like around, let's take the values. This is 7. This is 1 and this is 14. Okay. Our graph is going to look something like this. Right? Now, you can see that, again, what can we see? We can see quite clearly that so we can see once again, first thing that we have to realize is that all the enzymes in our body have an optimum temperature that is 37 degrees centigrade. Every single enzyme in our body has the same temperature, optimum temperature, that is 37 degrees centigrade. Okay. But just like temperature, just like enzymes can have a specific temperature, one second. So our body, just like our enzymes, all have an optimum temperature. Okay. So enzymes also have an optimum pH, meaning that there is a certain pH at which they will do their best possible function. Okay. At optimum pH, you will get the highest rate of reaction. If the pH is too high or too low, what will happen? The enzymes will get denatured. Once again, due to high pH or due to low pH, the enzymes are going to get denatured, meaning that the shape of active site will change. As the shape of active site is going to change, once again, the shape will no longer be complementary and enzyme cannot bind to substrate. Now, the interesting thing is that all our enzymes have especially in our human body, all the enzymes have the same optimum temperature. Okay, they all have the optimum temperature of 37 degrees centigrade, but there is no one optimum pH. Okay, for example, if we have enzyme, and Hajra, you should remember this, we have enzymes like pepsin. Pepsin is an enzyme that is found in the stomach. Okay, it's an enzyme. What is it? It's an enzyme. Where is it found? It's found in the stomach. Pepsin, because everybody knows what is inside our stomach, acid, right? We have acid inside our stomach. So, pepsin actually works at the optimum pH for pepsin is acidic. The optimum pH is acidic. It will work best around pH 1 or 2. On the other hand, we can have enzymes like trypsin. Trypsin is also an enzyme, just like pepsin. But trypsin is released in the small intestine. And I will teach you guys. Hajra has done it. Johara and Sarah, I will go over this with you guys, don't worry. So, trypsin is an enzyme that's released in small intestine, and the optimum pH for this enzyme is actually alkaline. So, see, you guys can notice in the difference in the graph. One enzyme is giving you high enzymatic activity even at low pH. pH 1, 2, it's giving you a very high enzymatic activity. And as the pH becomes more alkaline, the activity becomes lower and lower. Why? Because as the pH goes past the optimum temperature, whether it becomes lower than the optimum pH or higher than the optimum pH. Okay, the the and the main thing that we have to realize over here is this, that we have to realize over here is that the as the optimum pH is going to change, right? As the pH will change, it will become greater or larger than the optimum pH. Regardless of what happens, the enzyme will get denatured, the bonds will get disrupted, the shape of the active site will change, and the enzyme will no longer be able to fit into the substrate. So this is an important thing that we had to go ahead and we had to talk about. Are we good with everything that we've discussed so far? Does anybody have any confusion?
No.
Aljohara, Sarah, all good?
Yes.
Yes.
Okay. Now, the last thing that you guys need to know in this topic is, one second, we need to discuss about intracellular and extracellular enzymes. Okay. So I told you we have two types of enzymes. We have anabolic and catabolic. This is another category. Okay. So we can have intracellular and extracellular enzymes. What are intracellular enzymes? Let's start with them. Okay. Intracellular enzymes are those enzymes that are located in the cytoplasm of your cell. They are found in the cytoplasm of the cell. Okay. So, and they work inside the cell. So, they're going to work inside the cell. Okay. So, these are intracellular enzymes that are found in the cytoplasm of the cell. So, let me give you an example. An example that I can give you of this is related to the topic of immunity, which I'll go over with you guys the topic of immunity. But when I do the topic of immunity, we will study about your white blood cells. Okay. Your white blood cells, one of the functions of white blood cells is they eat the bacteria and then they break them down. Okay. So they have inside their cells, they have enzymes. Okay. They have protease enzyme. Okay. That protease enzyme is present inside the white blood cells. The white blood cells will eat the bacteria and they will use the enzymes to destroy them. So these are your intracellular enzymes. We can also have extracellular enzymes. Extracellular enzymes are released by the cells and they function outside the cell. Right? They are released by the cell. The cell will release them. They will go into, whatever areas around them, and it is going to break them down. A good example of this is are most of the topic of digestion. Okay, our, for example, our pancreatic enzymes, all the enzymes that are produced by the pancreas, they don't work in the pancreas, they don't stay in the pancreas, they are released from the pancreas into the small intestine, right? They are all released into the small intestine. Who makes them? The pancreas makes them. But where do they go? They are all released into the small intestine, all of them, right? So this is an example of extracellular enzymes. So our enzymes can be intracellular or they can be extracellular. Intracellular means that they'll work inside the cells, while extracellular means that they are going to work outside the cells. Okay, so that is all you guys need to know regarding your topic of enzymes. I hope nobody has any questions. Is everything all clear for everyone? If everyone is good with everything, then we're just going to take a look quickly at a question on the topic of enzymes. Let Give me 1 second. Let me share with you guys a question so we can see what kind of questions we get in the exam. All right, so our first question is saying the graph shows the effect of pH on the rate of enzyme catalyzed reaction. What explains the effect of pH on the reaction rate? So we have to figure out that which of the following options, we have four options. Which of them explains the effect of pH? Option number one, A says, as the pH increases from 5 to 7, the kinetic energy increases. That's why the rate of reaction increases. As the pH increases from 7 to 9, the frequency of enzyme and substrate collision increases. The shape of the active site changes as the pH increases from 7 to 9. Or D, the substrate denatures at pH 7. Now, let's go ahead and what do you guys think? What should be the correct answer over here? If everybody has an idea of what the correct answer should be, go ahead, let me know. Otherwise, you can also tell me which options you think are incorrect. Take a minute. You can guys can take a minute to think about it. Hajra, what do you think? Which of them should be the correct answer? Or which options do you think are incorrect?
I think it's not the first one.
Yeah, you're right. It's not the first one. Why is it not the first one? Okay, like, can you give me the logic behind it?
Um,
Think about it. That kinetic energy, they're saying that as the pH increases from 5 to 7, the kinetic energy increases. What does the kinetic energy depend upon? Right? Like for the kinetic energy to increase, what needs to change? If I want the kinetic energy to increase, what needs to change? What needs to become greater? The temperature, right? So if the because the thing is that kinetic energy increases, so where is that energy coming from, right? Like you must have studied in physics that energy is always conserved. So where is that energy coming from? If the kinetic energy is increasing, how is it increasing? So there has to be some kind of heat energy. Okay, so when the temperature will increase, that's when the kinetic energy will increase. Over here there is no rise in temperature, so kinetic energy will be the same. That's why D cannot be the answer. Hajra is absolutely correct. D cannot be the answer. So, Sarah, what do you think the answer can be? Or if you don't know the answer, then what option do you think is incorrect that this one cannot possibly be the answer?
I think C is the answer.
You think that the shape of the active site changes as the pH increases from 7 to 9. So, as the pH goes from 7 to 9, Sarah thinks C can possibly be the answer. Okay, let's note this down. Al Johara, any, what do you think? What do you think can be the answer over here?
C.
Al Johara also thinks that the answer should be option C. Okay, let's discuss, before we go over the final answer, um, let's discuss the options that we have. First of all, why not option D? Al Johara, why do you think it cannot be option D? Like, what's wrong with option D?
Because the enzyme works best at pH 7.
Yes, because pH 7 is the optimum temperature. So, most definitely, it is not going to get denatured. Absolutely correct. What about the second option, option B? Sarah, what do you think might be the reason why option B is not the correct answer? The second one.
Option B.
No, no. Option D is definitely last one is definitely incorrect, right? Because like Aljora just said that optimum pH 7 is the optimum pH. So at optimum pH, the enzymes will not be denatured. They are going to be working really, really good, right? So
Yeah.
I'm asking you about the second option, option B. Why do you think option B is incorrect? What's wrong with option B?
I think option B is also for temperature because if the kinetic energy
Yes.
increases, the collisions
Yes,
increase.
yes, exactly. This is also related to the concept of kinetic energy. Okay. This is again for the frequency to increase, the kinetic energy has to increase. Right over here, there's no change in kinetic energy. So the correct option we have is option C. The shape of the enzyme's active site changes as the pH increases from 7 to 9. That's why at 9, the enzyme activity is 0 because the shape of the active site has denatured between 7 to 9. All right. Okay, perfect. All right, guys, we're going to go ahead and stop it over here for today. And we will continue with the questions in the next class. I'm sorry, can you just stay behind for 1 minute?
Sure.